Bismillahirrahmanirrahim. Assalamu alaikum. Today we are going to start the series of lectures in which we will discuss the different types of drugs and the classes of drugs and their action on the body, and if they are present in the body itself, then what are their actions on the body, from where they are released, and what if we form the similar type of compound, what will be the structural activity and what is needed and what is not needed. Among these, we'll discuss hormones first. Hormones के बारे में एक बात जो आपको जहन में रखनी है वो ये कि majority of the hormones, especially the non-steroidal hormone, which अभी मैं आपको describe करूँगी which are कौन से होते हैं, जो non-steroidal hormones होते हैं, they are difficult to basically synthesize because they have a complex structure, and not only they have a complex structure, but the जो naturally present हैं in आपकी body के अंदर hormones या different bodies के अंदर living organisms के अंदर, so they are जो उनका पूरा structure है ना, that is important. The whole of the structure is important basically, and you cannot say there are certain points where you can change or where you can enhance the activity, and we'll discuss that also. But in majority cases, you cannot change the structure, or there are certain components which are very important, which should be present in those hormones for their activity. Okay, I'll not go with the screen, with the whole of the screen in my lecture, because I want to use my mouse as well. So I will not be using the whole of the screen, but the side screen view for my presentation. Okay, for classification of hormones on the basis of their nature, basically what type of nature they have, किस चीज से basically वो बने हैं, on that basis, the hormones are classified into two groups. These are proteinaceous groups, that is hormones, proteinaceous hormones. These are peptide hormones, and non-proteinaceous hormones, that are steroidal hormones. Okay, proteinaceous hormones and non-proteinaceous hormones, those which are basically protein based and those which are non-protein based. And if we see the chemical classification of hormones, we see there are two types. Number one is the steroidal types, and the second is the non-steroidal, that is the peptide hormones. The peptides या protein based hormones, वो कौन से होते हैं? These are insulin, glycogen वगैरह जो होते हैं, these are the peptide hormones, while amino acid derivatives, these are the thyroidal hormones. These are example of the amino acid hormones, amino acid derivative hormones, and miscellaneous में आते हैं prostaglandins and cytokinins. While in steroidal hormones, we have adrenal cortical hormones and then we have sex hormones. In sex hormones, we have different male and female sex hormones. In female sex hormones, we have estrogen and progesterone, while in male sex hormones, we have androgens and testosterones. Okay, first we'll discuss the proteinous hormones, which are basically protein-based. We all know that protein is an important element in our body, and protein as an integral part or integral element which takes part in many important and essential phenomena कह लें, या which is secretions कह लें, उसके लिए they are very important. ठीक है? Proteins are essentials to all living matters and perform numerous functions as cell components. They are also they function as cell components, and they are essential for all living matters. Fundamental cellular levels are catalyzed by protein called enzymes, and the one of the very important proteinous nature compounds are enzymes, basically, which are very important in catalyzing at different levels, which is the cellular level. While as other proteins serve as architectural constituents of protoplasm and cell membrane, they are basically the building block, how the building block of the structure of a compound. That's why they are called the architectural constituent, because they are basically what is the structure of a component or a part of a cell. That's why they are called architectural constituents. Although there are different hormones in the human body, they can be divided into these classes based on their chemical structure. These are lipid derived, which are usually the steroidal type of compounds. These the hormones, they are lipid derived, and there are amino acid derived or peptide hormones भी कहते हैं, ठीक है? Okay, amino acid derived hormones, वो कौन से होते हैं? The amino acid derived hormones are relatively small molecules derived from the amino acids tyrosine and tryptophan. So mostly they are derived from tyrosine and tryptophan. If a hormone is amino acid derivative, its chemical name will end in ene. Example of amino acid derived hormone include epinephrine and norepinephrine, which are synthesized in the medulla of adrenal glands, and thyroxine, which is produced by the thyroid gland, and melatonin produced by the pineal glands. While the peptide hormones, they have a little complicated structure, or I can say a more bigger structure. The structure of peptide hormone is that of a polypeptide chain, that is chain of amino acids. It has different chains of amino acids. The peptide hormones include molecules that are the peptide hormones include molecules that are short polypeptide chains, such as antidiuretic hormone and oxytocin, produced in the brain and released into the blood in the posterior pituitary gland. Peptide hormones में कौन-कौन से include होते हैं? They are short polypeptide chains, जैसे कि antidiuretic hormone, जैसे vasopressin है, oxytocin है, जो कि produce होता है brain के अंदर और pituitary gland के जरिए blood के अंदर आता है. This class also includes small proteins such as growth hormones produced by pituitary gland and large glycoproteins such as follicle stimulating hormone produced by this is also produced by pituitary gland. Okay, number one, which we will be discussing is insulin. You all know what is insulin. Insulin is basically a hormone which is created by your pancreas that controls the amount of glucose in your bloodstream. Basically, रेगुलेट करता है इंसुलिन किसको? बेसिकली इंसुलिन रेगुलेट करता है बॉडीज के डिफरेंट मैकेनिज्म्स को, जिसमें इंक्लूड होते हैं कार्बोहाइड्रेट, प्रोटीन और फैट की मेटाबॉलिज्म ऑल्सो, ठीक है? और इंसुलिन जिस वक्त सिंथेसाइज होती है ना, तो सिंथेसाइज होने से पहले जो जीन के अंदर इनकोडिंग जिस वक्त हो, जो जीन इनकोडिंग के जरिए जो इनकोडिंग के जरिए पता लगा है कि इंसुलिन जो होती है वो बेसिकली 110 अमाइनो एसिड उसके प्रिकर्सर होते हैं. ऑलमोस्ट कितने? 110 अमाइनो एसिड प्रिकर्सर होते हैं, जो कि फिर जिसको प्री प्रो इंसुलिन कहते हैं, जिनको ये प्री प्रो इंसुलिन 86 अमाइनो एसिड जो कि प्रो इंसुलिन के अंदर चेंज होती है, और ये प्रो इंसुलिन जो है वो क्लीव्ड होके इंसुलिन बनाती है, which is around 51 amino acids. Insulin is synthesized by the isle beta cell from a single chain. 86 amino acids polypeptide precursor, that is proinsulin, ठीक है? But this proinsulin is again formed from pre-proinsulin, जिसके अंदर 110 amino acids होते हैं तकरीबन. Proinsulin itself is synthesized in the polyribosome of the rough endoplasmic reticulum of the beta cells from an ever larger polypeptide precursor, which is pre-proinsulin. Insulin was the first protein for which a complete amino acid sequence was determined because it was a very important component or very important enzyme कह लें या very जो है वो था because it was insulin वो भी जब से ये determine हुआ था कि it is involved in this diabetes or diabetic patients के अंदर जो है इन्वॉल्वमेंट है तो इस वजह से इस पे बहुत काम हो रहा था और इट वाज ऑलमोस्ट इसको कहते हैं कि सबसे पहले जो है वो अगर सीक्वेंसिंग किसी भी कंपोनेंट की जो लार्ज पेप्टाइड हार्मोन्स थे उनकी की गई उनमें इंसुलिन वाज द फर्स्ट वन. Insulin has pronounced effect on the metabolism of carbohydrate, lipids, and proteins. This I have discussed. What are the different biological activities of insulin? Let us go through with these, although you have done it in numerous courses as well, but we'll go through again. Insulin enters the portal vein and about 50% is removed in its first passage through the liver. The plasma half-life in insulin is approximately 4 minutes. In addition to glucose, other substrates, that is amino acids, free fatty acids, and ketone bodies also can stimulate insulin secretion directly. Not only glucose, because insulin to secrete hoti hai after the addition of glucose in the blood. लेकिन सिर्फ यही stimulus काफी नहीं है. There are other stimulus जिसमें amino acid itself, free fatty acids और ketone bodies भी stimulate करती हैं insulin secretion को directly. इसके अलावा secretion and ACTH can directly stimulate insulin secretion. Secretin and glucagon and other related peptides can increase the secretion of insulin, whereas somatostatin inhibits its secretion. Elevated levels of circulating insulin decrease the number of insulin receptor on target surface and vice versa. Other factors that affect insulin binding to its receptor include pH, temperature, membrane lipid composition, and ionic strength. High glucose level also result in less water binding, less water being reabsorbed by the kidneys, causing high amounts of urine to be produced. This may result in dehydration. ये especially profound है in diabetic patients. Over time, high blood glucose level can cause nerve damage to the eyes and peripheral body tissues as well. Damage to the kidneys and cardiovascular system. And we while we have studied the diabetes mellitus, we have seen that patients who have prolonged history of diabetes, they have shown to damage their nerve damages, which lead to for the problems of eye, and they have other problems. There are other problems like kidney and cardiovascular system damages as well if this condition prolongs. Okay, now is the chemistry. जो इंसुलिन का स्ट्रक्चर है ना, इट्स अ कॉम्प्लिकेटेड स्ट्रक्चर एंड इसे डबल हेलिकल भी कह सकते हैं, बट इट्स नॉट अ डबल हेलिकल. बेसिकली जो स्ट्रक्चर है दे इट डू हैव टू टाइप्स ऑफ चेन्स, दो टाइप ऑफ चेन्स तो हैं, बट दे आर काइंड ऑफ ओवरलैप्ड टू ईच अदर इन अ वे कि वो एक जो है वो कॉम्प्लेक्स मॉलिक्यूल बनाती हैं. बट ऑफ कोर्स for the ease of remembering, I have okay, there are two parts of insulin chain. The polypeptide peptide contains about 51 amino acids. जैसे कि मैंने पहले describe किया कि they are altogether 51 amino acids arranged in two chains, that is A and B. The A chain contains about 21 amino acids, while the B chain contains about 30 amino acids. They are linked with disulfide bonds. There are two disulfide bonds which is present and which are basically a bridge between the two chains. Although the amino acid sequence of insulin varies among species, certain segments of the molecules are highly conserved, including the position of the three disulfide bonds. Means these disulfide, the three disulfide, sorry, they are not two, there are three disulfide bonds, and their position is very particular, and both ends of the A chain and the C terminal residue of the B chain, they are also very specific, and there is no. These similarities in the amino acid sequence of insulin lead to a three-dimensional conformation of insulin that is very similar among species. And insulin from one animal is likely biologically active in other species. Because of this similarity of amino acid sequence, we have a resemblance in the insulin among various species, various animal species, and including human insulin. That's why the insulin is isolated from different animals and used as a replacement for the insulin, which for the patients, human patients. Insulin molecules have a tendency to form dimers in solutions due to hydrogen bonding between the C terminal of B chains. Additionally, in the presence of zinc ion, insulin dimers associate into hexamers. This is its intrinsic property, जिसे कहना चाहिए इसकी है ये कि इंसुलिन जो है वो डाइमर्स बनाती है, यानी दो मॉलिक्यूल्स इकट्ठा होके जो है वो सोल्यूशन के अंदर वो डाइमर्स बना लेते हैं, क्योंकि इनके दरमियान बहुत अच्छी हाइड्रोजन बॉन्डिंग हो सकती है. दैट्स वाई इसकी वजह से ये यूजुअली डाइमर्स बनाते हैं. और अगर इसमें जिंक आयन की भी अगर वो शामिल हो जाए तो उसके साथ साथ डाइमर्स के साथ साथ हेक्सामर्स भी बनाते हैं ये. ठीक है, दिस इज द टेंडेंसी. अब थोड़ा इसकी स्ट्रक्चर एक्टिविटी रिलेशनशिप की तरफ आते हैं. अब ये जो सारा स्ट्रक्चर है ना, द सीक्वेंस विच इज ये ए चेन का और बी चेन का जो सीक्वेंस है, ये आपने थोड़ा सा याद रखना है कि इसका सीक्वेंस क्या है और इसके सीक्वेंस जो है उसकी डिफरेंशिएशन की वजह से क्या क्या चेंजेस आते हैं. The natural sources of insulin are mainly porcine and bovine pancreatic tissues. In comparison to synthetic insulin, natural is cheaper and is readily obtained in large amounts. SAR studies carried out on insulin reveal the following points, which are: number one, there exists a definite relationship between the conformation of insulin molecule and its activity. जो भी conformation है insulin की, जो भी मैंने आपको दिखाई A और B chains की जो conformation है, इसका पूरा कॉन्फर्मेशन की जो है वो इसकी एक्टिविटी के साथ एक खास रिलेशनशिप है. एनी अटेम्प्ट टू चेंज दिस कॉन्फर्मेशन लीड्स टू डिक्रीज और इंक्रीज इन एक्टिविटी. Decrease in activity only. Disturbance in the sequence of amino acid chain in chain A reduces the activity. If you disturb the sequence of amino acids in chain A, it reduces the activity, while the amino acids from 1 to 6 and 28 to 30 can be removed from chain B without significantly affecting the activity. आप A chain को के sequence को खराब करेंगे तो activity lose हो जाएगी, while आप B के अंदर there are certain amino acids which you can totally remove, and without those amino acids you still get the activity. Reduction of either chain abolishes the activity. If you try to reduce the whole of the chain, यानी A हटा दें या B हटा दें, तो आपको activity बिल्कुल ही नहीं मिलेगी. Any modification of the side chain carboxylic group or the tyrosine residue tends to decrease the activity. और ये भी है, याद रखिएगा कि दोनों चेन्स में अगर आप जो है साइड चेन के अंदर कोई चेंजेस लाते हैं तो भी एक्टिविटी लूज हो जाएगी. It may be concluded that chemical modification in the insulin structure failed to potentiate the hormone activity. It may be concluded that chemical modification in the insulin structure failed to potentiate the hormone activity. Some analogs have been prepared but are of limited use to study insulin. Basically, जो modification की गई ना, उस मॉडिफिकेशन के जरिए जब उसको हार्मोन को मतलब चेक किया गया तो उसके अंदर कोई खातिर खा पोटेंशिएशन नहीं हुई, बिकॉज क्योंकि देयर मैंने आपको पहले भी हमेशा ये समझाया है कि स्ट्रक्चर के जो जितने फंक्शनल ग्रुप या जितने भी कंपोनेंट्स हैं जो प्रेजेंट हैं, ये सिर्फ एक्टिविटी के हवाले से इंपॉर्टेंट नहीं होते कि वो एक्टिविटी कैसे प्रोड्यूस करें, बल्कि वो उसके सर्टन टाइम्स उसकी बेटर रिसेप्टर बाइंडिंग और उसके लिए भी इंपॉर्टेंट होते हैं। इस वजह से जो खातिर खा आपको जो है वो अच्छे नतीजे नहीं मिले आपको इंसुलिन के हवाले से, क्योंकि इंसुलिन के रिसेप्टर बाइंडिंग भी बहुत इंपॉर्टेंट है फॉर इंसुलिन की इफेक्ट के लिए. The therapeutic importance of zinc and chromium in the treatment of diabetes is recently reviewed. Zinc, being an integral part of the insulin structure, is also found to affect carbohydrate metabolism, while chromium activates insulin and its receptor site and is found to increase both the affinity and intrinsic activity of insulin. जिंक और क्रोमियम की मौजूदगी में जब डायबिटीज के अंदर इसको इस्तेमाल किया गया तो ये it was found that it increase the affinity and intrinsic activity of insulin. Okay. अब हम थोड़ा चेन के हिसाब से करेंगे कि अगर आप चेन बी चेन के हवाले से बात करेंगे कि अगर उसके अंदर चेंजेस लाएंगे या कहाँ ला सकते हैं, और ए चेन के हवाले से. बी चेन के हवाले से अगर हम बात करें, several residues of the B chain are not required for full bioactivity. कुछ इसमें अमीनो एसिड्स ऐसे हैं जो कि रिक्वायर्ड नहीं होते. The first 4 to 6 and the last few residues are dispensable. जो first 4 और जो last 3 residues हैं वो आप हटा सकते हैं. As further residues from COH terminus that are dispensable, यानी जो कार्बोक्सिलिक ग्रुप है उससे मतलब के दूरी पे जो है वो B28 और जो 30 है these are dispensable, जबकि B27 और 26 they can also be omitted. The biological activity decreases gradually. लेकिन अगर आप ये डिसहेप्टापेप्टाइड जो है इंसुलिन, ये बेसिकली क्या है कि इसकी जो लाइसिन है उसको कम्प्लीटली जो है वो आर्जिनिन के साथ जो है वो रिप्लेस कर दिया गया हो ना, तो ऐसे में कह रहे हैं कि बायोलॉजिकल एक्टिविटी बिल्कुल ही इनएक्टिव हो जाता है. ठीक है. Residues, that is 24 to 26, occupy important parts of the receptor binding area of the protein, so you cannot change the 24 and the 26 amino acid. Okay, in chain A, the chain A cannot be shortened. Omission of A1 glycine residues, the hormonal potency to less than 1%, and omission of A21 asparagine abolishes the activity. However, replacement of A21 asparagine by alanine preserves full activity. The A1 guanidinated insulin is the most active derivative. A positive charge increase for A1 lyse and lysine and arginine insulin, and a negative charge decreases the activity for A1 glutamate या glutamic acid derivative.